Okay, welcome everybody. Please take a seat here in the room in Berlin. I would like to welcome you to our first edition of this year of the Digital Sciences for Drug Discovery seminar series. And I'm very excited that we're going to have two speakers here today in person. And while people are still dialing in, I'd like to make a couple of comments on our series. If I can move the slides. Okay. So the aim of this series is really to bring everybody interested in the topic of digital science for drug discovery together across disciplines, across industry and academia. And well, it's scientific talks, but it's also socializing. So we're going to meet later here in person in Berlin, and there will also be a virtual networking event on Zoom. So stay on after the talk. And who is we? Well, that's myself, Andrea Volkhammer, Andreas Bender, and Gerhard Wolber, who you will see also in the later on in this seminar. And with that, some last housekeeping remarks. So we're going to publish the next event date on our webpage that you can see here, both as a QR code and as a link. But we also advertise it via social media. We will record parts of this meeting, and we will make recordings available whenever that's possible. And well, you can have, of course, questions here in the room, but you can also, alongside the talks, type your questions into the chat window, and we'll call you to phrase your, to put your question then orally later on. Yes, and with that, I would like to ask Gerhard on stage to introduce our first speaker. Gerhard, please.
Switch microphones. Okay, thanks. Thanks, Clara, for introducing and making these general remarks. Just switch to microphones very quickly. Okay, so it's an honor for me. So for me also, welcome to everybody, especially from the FU. We do have about 200 registrations for the meeting, and I don't see how many are dialed in actually, but it's quite a lot. So yeah, thanks to just multitasking for the technical stuff at the moment. So if there is anything, any technical issue, please write to the chat anytime. We have Christina here taking care of the technical part and letting us know if anything is going wrong. So it's a big honor for me to introduce our first speaker, John Chodera. It's a big chance that he's really here in Berlin this week, and Andrea organized that he will join us for this event here in presence in person. John did his PhD in biophysics at USASF. Most of you will probably know him, but I just shortly introduced him anyway. And then did a postdoc with Vijay Pandey from 2006 to 2008, and then became independent researcher in Berkeley before he moved to the Sloan Kettering Institute, where he still is. He became associate professor there in 2018, and also his connection to Berlin is that he became a BIH visiting fellow from 2019 to 2021, I suppose, but this has now been prolonged, right? Yeah, which we're very happy about. So we're very much looking forward to your talk. John.
Thanks so much, Gerhard and Clara, and everyone for inviting me today. I'm super excited to be here. I love Berlin. I've been coming here, I realize now, for 20 years, and the bus numbers haven't changed. It's great. So I'm, like Gerhard said, I'm at the Memorial Sloan Kettering Cancer Center. It's a cancer center that actually treats cancer patients. 90% of it is the hospital, but 10% of it is the Sloan Kettering Institute Basic Science Research Enterprise, where I am in the computational systems biology program. We've got a bunch of wonderful faculty who do experiments and computation, where the experiments often feed the computational models. I'm right next to Rockefeller University in the Upper East Side of New York, and all of our graduate students come from the Weill Cornell Graduate School of Medical Sciences. So we have something like 1,000 faculty all interested in human health and biology just on this one street corner. So it's a fantastic place to work. And if you're looking for a faculty position, we're hiring every year, so please apply, and we'd love to have you. So my lab looks at how to build models for areas of drug discovery where there's not really good predictive models yet, where we still have needs for more accurate models or models to predict other properties. To do this, our main focus is using good statistical mechanics and integrating physical modeling and machine learning to make good predictions, but we also sometimes need to gather data. So we also have a robotic wet lab, which you can see at the upper right. This is our robotic undergraduate that moves plates around and does experiments for us. We're also trying to use things like the AWS for wet lab experiments, which is Stratios, previously Transcriptic. They have all of these great things, which are very much like our robot, but you can queue things up in the cloud and they'll run experiments for you, or even little almost disposable robots. These Opentrons are handcrafted in Brooklyn by artisanal handcrafters using 3D printer parts, but they're so cheap that they're almost disposable. So you can get these and program them in Python to do experiments. These are things we use to collect a lot of data for binding affinities primarily that feed our models. We use molecular mechanics simulations and molecular simulations accelerated on GPUs. And then we run a lot of stuff on Folding at Home, this worldwide distributed computing system that recently was the first distributed computing platform to cross the exaflops barrier. So there's a lot of folks that are primarily gamers, and when they're not playing games, they contribute their GPU computing to biomolecular simulation modeling. We're very thankful for that. We work on a wide variety of problems, and I'll talk about some of the focus of what we're trying to tackle now in kinase inhibition, where it's really important for cancer, of course, because these are logic gates that control whether or not the cell is supposed to proliferate. And if they go wrong, if they're dysregulated, then they can cause unchecked cell proliferation. You end up with a tumor or leukemia that ultimately kills you unless you intervene. And so we're trying to design targeted therapies that are very selective, very potent, but are selective for one kinase or a couple of kinases out of 500 that all share the same kind of ATP binding site. So it's a challenging problem that I'll get into a little bit about how we're trying to tackle that. And as well as the other aspects of that in just a moment. We're also interested in how we can target new therapeutics specifically to cells. And then all sorts of exciting stuff is going on in cancer immunotherapy right now with modeling this immune synapse between T cell receptors and cells that present cancer peptides on their surfaces. And then there's a whole world of antiviral discovery I'm not going to have a chance to talk about today, where we've been working on discovery of antivirals for the current and future pandemics. So cancer is obviously an important disease to worry about, at least in the United States. It's the #2 killer, and it touches many millions of lives. The number of people who have lived with some history of cancer in the United States alone is something like 17 million people, which is a lot of people that have to worry about the potential for relapse. And while we've made a lot of strides in improving the state of cancer therapy for many kinds of cancers, there's still many of these for which we really haven't improved on the survival in the last 20 or 30 years. And that's really unfortunate. It's like really a crapshoot where there's not a lot of good therapies for some of these kinds of cancers. And the kinds of hopeful ways to target cancer that we've been focusing on are using these targeted small molecules that go after specific kinases that are dysregulated in many types of cancers. Just to give you an idea of what this looks like, you might have a particular kinase. This is the kinase domain that does the phosphorylation. And one particular molecular event, a fusion of the breakpoint cluster that displaces the regulatory region of Abl kinase, can lead to unchecked white cell proliferation and then ultimately a leukemia that kills you. But this miracle drug imatinib was approved by the FDA in 2001, shuts down imatinib relatively specifically, even though it targets the ATP binding site that's shared by all kinases. And by doing so, you can effectively cure many of these patients. And so it was like this magic bullet that we were hoping to replicate for many other kinds of cancers. I think there's now been 72 small molecule kinase inhibitors that have been approved by the FDA since then, and it's rapidly growing, but it's still a really challenging area because the number of kinases that we've targeted is still very small compared to the number of kinases that are dysregulated in different types of disease. And we are seeing a decent number of these, but it's far fewer than we actually need to actually target different therapeutics. And you'll see that many of these under clinical investigation, there you have many inhibitors targeting the same kinase, so we haven't really extended our repertoire of therapeutics to the point where we can really have impact on all of these types of cancers yet. And then it gets worse, which is that after you treat a patient with a therapeutic for a while, you either are selecting cells that might have resistance pre-existing, or you might be having resistance generated during the time that you're treating, because if you can't completely eliminate the tumors, then you have repair deficient cells that are trying to make mutations and get around your drug, so that if a single mutant, for example, can ablate inhibitor binding while still carrying out kinase activity, then it's going to be bad news, and you'll have a tumor that ultimately is just drug resistant, and the patient will ultimately die. So there's many mechanisms by which this can happen, and we've been trying to come up with a mechanistic-based approach where we can predict and actually tackle these different mechanisms of how we can address these. One of them is obviously you can have a mutation which knocks down the inhibitor affinity directly, or it can modulate the activity for ATP or the substrate and increase the activity otherwise, so you need more drug to actually counter that, or it could alter the conformational populations in ways that might disfavor drug binding and favor activation of the kinase to even drive kinase progression further. And so coming up with mechanistic ways to predict these would be very valuable because we would be able to make useful predictions and design things that could address them, but it's still very challenging. So I'll get at some of how we're trying to do this. The other problem we have is that some of the mutations that we see in patients, these are the counts of mutations that we might see in one of the often dysregulated kinases, EGFR, are often wildly different. So some of them are highly recurrent, where there are good drugs that treat L858R perhaps, but most of the other mutations, we don't even have any biochemical evidence about what this does. Is it an activating mutation that might drive disease? Is it something that might be resistant to therapy? Which therapeutics might actually work the best? Are there unmet needs for developing new therapeutics where we'd like to actually address all of these? This is something where we don't quite know what's going on here. And if you look at the fraction of time you've seen these mutations, 87% of them have been seen less than 10 times. So we really have no idea, no, certainly not clinically, and maybe not even biophysically or biochemically, what's going on with those particular mutants. So how does one actually tackle a drug discovery project in this area? I'm just going to take you, some of you are probably very familiar with this process, but for those of you who aren't, when you start a discovery program, you have to start with some statement of your objectives. What is it you're trying to achieve? And so I had my first experience riding along in a discovery program as part of the COVID moonshot as we were doing antiviral discovery for SARS-CoV-2. That antiviral is now on its way to hopefully an IND-like approval, thanks to the DNDi and the Wellcome Trust. The Drugs for Neglected Disease Initiative is leading that effort now to get into clinical trials in a patent-free, low-cost way. But you have to have some statement of your objectives and say that I want to inhibit my particular target. In that case, it was the protease of the main viral protease for SARS-CoV-2. You also have to get into cells, right? In order to actually permeate the cells, you have to get through the membrane. It has to be soluble enough. There's other considerations involved as well. If you want it to be orally administered, then it has to have a decent solubility and different dissolution characteristics. You also have to have it stick around in high enough concentrations free in the plasma to actually do the effect you intend. And then there's a whole host of other safety issues that you have to carry about, many of which are maybe mechanistically informed, where you can't inhibit these cytochrome P450s to too high of a degree, but you also have other things like you just don't want dogs to drop dead. And that's very much harder to predict in some cases. In order to achieve those goals, you generally construct this set of assays, this cascade that allows you to address these properties. You have a measurement that pairs with each of your objectives, one to one, and then some way of stratifying them into different progressions so that you don't, like some of these assays involve animal experiments, which are very expensive, ethically, have ethical considerations around them and really take a long time to do. So you don't want to have to send so many molecules through these more expensive experiments, and you'll stage the easier experiments first in your first tier and check things like, does it do the thing to the target and does it get into cells? Is it likely to have other problems that cause issues with developability? And we're in a position where we have much more complexity with dealing with kinase inhibitors, of course, because we also have to worry about not only one target, but a whole spectrum of different nearly identical active sites in kinases where we have to achieve the selectivity or polypharmacology we might want in case we have to hit two different kinases to have a desired effect. And then we also have to look at mutant selectivity spectrum. So if there are multiple mutations that we know are driver mutations for this particular cancer in this kinase, we might want to hit all of them, but spare the wild type in order to prevent, to have a better therapeutic index. We want to spare the normal wild type cells that are unaffected by cancer if at all possible. So it's a very complex set of design objectives. Now there's hope for this, that we could eventually engineer enough different models, mechanistic models or machine learning models or target-based models, and I'll talk about one of those classes today, in order to actually have an impact on how to drive these programs in an informed way. And so for each of these assays, ideally you'd have a surrogate that you could use to predict and evaluate many different molecular designs as you're making decisions through the design, make, test, analyze cycle. So most of the drug discovery, early discovery phase time is spent in this cycle where you're taking all of the knowledge you've accumulated so far, looking at your best molecules so far, designing new molecules that you then evaluate and prioritize for synthesis, making them and assessing them, and then pushing them through this assay cascade and hoping that you can get some of the molecules at least to figure out if they do the right thing in some sort of organismal model or cellular model, and then ultimately nominate a preclinical candidate that you're willing to actually develop potentially into human clinical trials. That's super expensive, so you have to be very sure that you're meeting your objectives there. So the cool thing is that, at least for target-based properties, which are usually the most challenging, like in this case, a lot of the other properties are common to the molecule. So you can pool molecules from all of your discovery programs and make potentially decent global models that you could learn from. But the target-dependent properties, where you have to predict something for this specific kinase in this particular mutation, are often extremely challenging. And so we like to use structure, or think about how we can use structure to improve our success rates in making useful predictions. And the cool thing is that we're in this amazing place right now where the last decade has seen more than 100,000 new structures available for us through the protein data bank. And of course, the tremendous excitement around AlphaFold has meant that we now have a much better way, much more facile way of extending that radius of comparative modeling using things like AlphaFold to predict structures of individual proteins or even protein complexes. Much of the kind of activity that you often see these models trying to make target-based predictions in is when you might have already a lead compound like this in the upper right, and you're trying to figure out, if I have a synthetic route already worked out, in the last step, I can substitute anything from a large building block library that's compatible with this reaction. In this case, for making this compound, installing that last group, in the last step, I can make a ton of the intermediate and then pick anything from the 5,000 compounds building block library at NMROps, for example, and make a bunch of compounds where a good chemist can make maybe a dozen compounds per chemist per week. If you had to do something else and nominate completely different compounds that required completely different synthetic routes, it would be very hard to debug and optimize those routes, and you would really reduce the productivity of molecules that you could test per week. And so what you'd like to do is to make some informed predictions about how these will impact the affinity, the selectivity, and the mutational effects for that target. And so we practice a technique and have been working on developing these alchemical free energy calculations for a long time. Some of you have probably used them or know what these are. Essentially, what you're trying to do is to predict something that's related to a free energy of binding, a free energy of mutation, a free energy of difference between two different ligands. And you do this by, instead of trying to do the direct thing of dissociating the ligand and looking at what the impact on the free energy is, which is really hard to do because it would require a lot of association and dissociation events that would just take unreasonably long timescales to get statistical precision. You instead transmute the ligand into something else, and you do it in a way that doesn't have to exist in chemistry. So by turning the ligand off through these weird intermediate states, as I show here, you discharge it or turn off its sterics, or turning one ligand into another ligand through a series of stages that can't exist in chemistry, you can get a rigorous free energy estimate much more efficiently, in orders of magnitude less time. And there's lots of folks that have contributed to how to do this efficiently. It also has some really nice overlap with other statistical machine learning methods, so that the students who work on this can go and put a 1 or a 2 in front of their salaries if they go off into industry and machine learning. It's a really nice technique for a variety of reasons, but it also is something that has enough generality that could be extended to a lot of different kind of target-dependent properties. And so we've been working for a long time on optimizing its ability to drive affinity or potency for a single target. This is sort of the thing that Schrodinger has really pushed within drug discovery, but of course there's folks like Clara who have contributed pivotal science to actually making this practical early on. Also, it turns out you can use these techniques to drive selectivity. So in work we've done with Schrodinger and other folks have assessed is what's the error you make in trying to predict the affinity to 2 different very related binding sites. And it turns out there's a lot of correlation in the errors you make in force field interactions that kind of cancel in those cases where you might end up in fortuitous cases where there's a cancellation of errors that leads to better prediction accuracy than in predicting affinities alone. And finally, there's a lot of exciting work going on in terms of predicting the impact of mutants on drug resistance. We have a fun collaboration that's still ongoing to try to quantify how well these methods can predict the impact of point mutations on small molecules. But it's a really promising area because you're just changing a few atoms and trying to assess what the impact is on binding. There's also been some fantastic work from folks like Bert de Groot on predicting the impact of point mutations on thermostability, which could be tremendously important for engineering biotherapeutics or antibodies. And then there's also a host of other properties where these are not necessarily target specific, but useful ways in which we can use these calculations to maybe say something useful about all of these other properties for the target candidate profile. In fact, there's a lot of exciting structures coming out for toxicity targets right now, thanks to cryo-EM. And some of these might be tractable. For example, you don't want to inhibit hERG because if you did block it in some way, you might cause terrible cardiac toxicity events. That would be very bad. And so, ultimately it comes down to questions like these methods can allow us to address what happens if we change a few atoms and figure out how to predict what the impact is on affinity or selectivity through looking at the impact of changing the molecule on how it binds a kinase, or how we can look at the impact of changing the mutation, the kinase, on the affinity for the actual drug. Now, the problem is that all of these alchemical free energy calculations depend upon some model of the interactions between the species. And what's surprising is that even though these are very simple models, we seem to be able to do a reasonably good job of modeling association of a small molecule with its target. There's a wonderful simulation of dasatinib binding to SARC from the Desres folks. But ultimately, these things have extremely simple structure. We throw away all of the quantum chemistry, and we model all of the valence terms, local valence terms, with low-order Taylor expansions, and then low-order Fourier expansions to model local interactions. And then we do some decent job of modeling electrostatics, but using fixed point charges instead of representing the polarizable clouds of electrons. We have some idea of what the dispersion looks like at infinite separation. It goes like 1/6. And then we square that because it was fast on a PDP-11 in 1983. And that's basically it. That's what we've been stuck with for the last 40 or so years. And it's surprising to me that we've been stuck here, and B, that these models work at all. But we are where we are. And the magic has been in how you come up with these parameters in a way that's somewhat generalizably applicable to a large variety of systems. And so the way you come up with these parameters is to take a series of graduate students and feed them through the meat grinder, and hope that some parameter set that people actually use and has some impact on drug discovery comes out through the end. If you wanted to simulate a system that was reasonably complex, so biology has lots of stuff in it, and you might need to simulate something that includes proteins along with some water and ions. It might have a small molecule that you're trying to design or look at the impact of a mutation on. Sometimes you have nucleic acids. There's a lot of interest in drugging those, or you have protein nucleic acid complexes. It might be associated with the lipid membrane, and often things have sugars on them or have carbohydrates associated in some way. We learned the importance of that in modeling the spike protein recently, for example, for SARS-CoV-2. Turns out this is the list of references that correspond to the force field components that you need to use together from the Amber manual. And it's great, it's a huge amount of pioneering work that's gone into these models. But if you add them all up, you get something like 100 human years have gone into just the force field that you're using. Right? It's a huge amount of human effort. And in order to update it, it makes it very difficult because all of these have been optimized independently and are meant to work together, but it's very hard to work them, to use them. It's very hard to ensure that they're consistently optimized if you're trying to update something. And just to give you an idea of what that looks like, this is the small molecule force field GAF1, which was finished in 1999 when I started graduate school. I was actually at Jun May's talk in the Coleman group when he was talking about finishing up the optimization, getting this done. This is sort of the variety of chemical structure moieties that were represented in the force field. If your molecule doesn't look like this, it's not going to do a good job. And if you need to add more atom types, tough luck. It's just very difficult to extend it because there's no fitting code that's released that publicly. And if you do need to make a new type, you have to make sure to duplicate all of the other parameters that are associated with the type in a completely error-free way using this hierarchical typing approach. And that has not happened in the past. So there's easy ways to introduce numerous errors if you do this wrong. This is very different from what we've seen in machine learning, right? Where we have really powerful machine learning frameworks that allow you to do exciting research with very compact expressive language. I'll just take you through this older example from the TensorFlow Keras, so TensorFlow 1 example, just because I think it's a really good example of what we would hope to achieve. You basically take a data set, in this case, it's the MNIST digits data set that's been curated from someone else, by someone else using best practices. So it's a established community data set, best practices curation. You split it into train and test. You define a new model architecture using a composition of really powerful abstractions because you can chain these objects together in a way that creates something new using a very minimal language. And then you take some best practices for how you fit it, how you what's your target objective for fitting and how you assess it. And then you say, I'm going to fit it, and then I'm going to use it. That's it. It's a whole research project, right? In just a few lines of code. But we can't really do that in our field, right? What you'd like to do is the same kind of thing where I compose a model together from different physical and other kind of learnable forms and use best practices data sets that somebody else has curated and then put it all together using best practices and actually use it. And then if more data comes in, you'd like to use that too. We're not there yet, but we're trying to get there through various approaches. So one of them is through the Open Force Field initiative, which is Clara is foundational in helping us launch. It's one of these open science pre-competitive efforts to try to build software infrastructure as well as data infrastructure for building next generation open force fields. So not only does it provide the tools to build and apply these force fields, but also the data sets that other people could use to evaluate or improve on these force fields, and then is using best practices established by the community to build and release new small molecule force fields, but also coming soon is biomolecular force fields as well. And everything is done in the open with open science, with feedback from others appreciated. And so this effort has been really useful in small molecule binding affinities, for example, even through the first few generations, going from where we were in 1999 for specific targets, it's really improved the accuracy. And now we're on Open Force Field 2 and looking towards Open Force Field 3 to be released pretty soon. But fundamentally, all of these things are limited by this idea that you have some types. These are types that we assign to either atoms or different interactions. We have, we might start off with each element might have a different type, but at some point you have to realize that carbon and aromatic ring might be different from a carbon and aliphatic chain, so you need a different type. And you could get more and more complex in these typings, but then you don't know where to stop. Like you could get easily overfit your data by just making more and more types, because the type encodes everything you'll ever need to know about that molecular context in order to apply a parameter to it. We would like to avoid that and come up with a different way to do this. So rethinking this, Josh Fass, a talented graduate student in the lab who's now at Relay Therapeutics, and Yuanqing Wang, another talented graduate student who is now going to be a fellow at NYU, came up with this idea of, well, let's learn not a discrete way of representing each atom or each type, but a continuous representation. And to do this, let's use a graph convolutional network where I can use a machine learning approach that knows about chemical equivariances. That is, if I have two different carbons that are identical in a chemical sense, like exchangeable without any change in their properties, then the graph convolutional network will also assume, or will also ensure that you end up with labels that are applied to those that are exchangeable as well. And these are like neural networks that are stacked in the same type of topology as your molecule, or you could think of it as passing messages along your bonds and applying nonlinear functions to learn about the molecular context several bonds away and how each carbon is slightly different from each other carbon if they're not chemically equivalent. And the cool thing with this is that you can apply this graph network and end up with a continuous embedding vector for each different atom, and that tells you something different about how that atom is different from others. And then you can combine them in interesting ways in order to get a vector that represents, say, a specific torsion. But you have to worry that a torsion, IJKL, is the same as LKJI, right? They have to be symmetric going forward and reverse. So there's a way of pooling these in a way that's symmetry preserving, that's also learnable with the original typing stage. And then you can come up with these modules that are fully independent of each other, but use these different embeddings in order to predict the force field parameters directly. So we could take any molecule, including a protein, perhaps, feed it in and come up with a way of predicting all the parameters to compute in a normal molecular mechanics simulation, the energy, forces, trajectories, et cetera. And you can use any loss function you want to fit this, whatever properties you want to fit to. And it's modular enough that if you want to come up with another way of representing polarizability, for example, you could just tack on another module and use whatever atom bond angular torsion property you want or feature vector you want to compute with it. So what that means is that this is the entire script for parameterizing a force field. You can do it in a day. Using quantum chemical data, for example. The cool thing about this is that it seems to do reasonably well. So if you feed it some quantum chemical data, you might do different experiments to ask different questions about how well it does. So let me just take you through a couple of these examples. First of all, you might ask, what happens if I really mine the space well? If I have a lot of quantum chemical data on different conformational energetics, and these are optimization data sets, so I enumerate conformers and then minimize them and use all of the snapshots that I generate there. I just have carbons, oxygens, hydrogens, and I've just have different isomers of them in different ways, and enough of them to really cover this space. And it looks like it does a decently good job, a little bit better than some of the molecular mechanics force fields, the traditional ones like GAF from Amber or the open force fields. This might have to do with just having a better representation of the differences between these different types, but it really becomes pronounced when you look at on the same data set that Open Force Field 1.2 is trained on. So this error, 2.17 kcals per mole, is the error on its own training set, but it's limited by the fact that it has these discrete types. Whereas if we go to this continuous way of representing atomic types and allowing it all to blend together, even on the test set, it does much better in representing this span of molecules. There's our little fragments of drug-like molecules that challenged industry in terms of trying to come up with good torsion drives, et cetera. You can also go to more interesting things like heterocycles that might be the scaffold to future drugs. Some of these are admittedly very crazy. Nobody would actually make some of these, but they are believed to be, potentially future molecules that could be the scaffolds of new drugs. If you look at the error for traditional force fields, it's something like 8 kcals per mole, and you're asking, like, how can it possibly be so large? Well, it turns out if you go looking at some of the largest outliers, you can see that you end up with compounds like this, which are not aromatic at all, in fact, that happen to end up in the set. And quantum chemistry correctly determines that these nitrogens should be planar. But just because a human has never thought of, like, I should assign a special atom type to this weird kind of nitrogen that nobody's probably going to make in this case, I just don't have a good way of representing it. But simply by showing it a few examples, it learns that it's different from other types of nitrogens in that way. So I think it's a useful illustration of how it gets into the cycle that we would want, where if we find failures, we just show it more data and it learns from those failures to improve itself. And we can show it peptides too. There's no reason we can't show it peptides as well as small molecules. You've got this PepConf set where it seems to do better than Amber, which is crazy because it's, this is decades of optimization, high-level quantum chemistry, combining things together with curated library charges. And this is none of that. It's just training in a few hours. And then you could start to do things like using both small molecule and peptide quantum chemical data to reparameterize an entire self-consistent force field, and then see that you have small molecule and a protein at the same time with all the parameters coming from this approach, espaloma, except for the water. We're still using tip3p water, which I'm embarrassed to admit at this time. But it seems to be very stable. And so the next thing you might ask is, well, how well does it do for a binding free energy for a kinase inhibitor? And the cool thing is that we're able to only apply the parameters to the ligand in this case. We don't quite have our workflow able to use the parameters for the protein yet, but it seems to do quite a lot better than the traditional force fields. So it's improved the error to about half a kcal per mole RMSE. So it doesn't do any worse, even though it hasn't seen any of this data yet. It's just trained on some small molecule data and some protein data. Now, this is an embarrassingly small amount of small molecule data. It's only 792 molecules. So we're trying to increase the sizes of data sets that we have to parameterize these to go further for next generations of this. But I think it's an interesting, compelling idea. And just to show you that you don't have to just use quantum chemical energetics, you can also use free energies if you want. Now, in principle, you'd have to use simulations in the loop if you're training to a free energy because it's an ensemble derived property, or if you want to do densities. But in this case, just to show off how this works, you can actually compute the free energy of hydration free energy of small molecule by running a vacuum simulation and then reweighting to the parameter sets with a new set of parameters. And you can still get just using the exponential average, the logarithm of that, you can get a really robust free energy estimate for vacuum molecules. And so you can use that reweighting and its derivatives in order to optimize this and show that you can actually reduce the error for an implicit solvent model to greatly over just by using a free energy instead of just quantum chemistry. So what we'd ultimately like to do, though, is be free of these Taylor order expansions from these early models that we've been stuck with since the 1980s, right? There's a whole new generation of exciting machine learning potentials. I've been calling them QML, but there's other names for them: machine learning potentials, neural network potentials. One of the more popular ones comes from Ola Sisiev and Adrian Reutberg, where you have a bunch of features that you use to featureize atoms in space. You have multi-body terms. You push these features for each environment around an atom through these neural networks, which can learn the contributions to the energy. You can get extraordinary reproduction of quantum chemical energies from this. And so you might ask, well, okay, if I throw away these crappy molecular mechanics force fields, at least for part of the system, let's do that for the ligand intramolecular energetics. So I can better model the intramolecular interactions and strain energies, and then add back the machine learning potential. How much does that get me? Because that's avoiding computing the whole system with machine learning potentials. They're really expensive right now, something like 15,000 times more expensive than MM alone if you just do them the standard way. So the cool thing is that you could do this by doing a normal MM free energy calculation, which we do all the time, And then you can correct what would happen if I perturb my end states of the ligand in complex and the ligand in solution into these ML-MM hybrid systems. You could do that at the same time even. You don't have to do too much. You just run the simulation and then you either switch the parameters with non-equilibrium or you use replica exchange or something like that. You can also, again, find, using the same system here, an increase in accuracy this way. In fact, we seem to be running into this wall of about half a kcal per mole, no matter what we're doing to improve the fit for this system. So I wonder if we're running close to the experimental error. But it seems to be very compelling that we might be able to do better than just MM for at least part of the system. And what's exciting about this is that many of these inhibitors have conjugated systems or hyperconjugation throughout the molecule. These are amide bonds where it has a little bit of double bond character, and it would make sense there's some correlation between the R groups that we have over here and this torsion and this torsion over here. In this case, we're substituting R groups on this side of the molecule for this series. And what you see that's missing in the MM is that you have a coupling between the torsions in the ML state as well as shifts in these distributions of torsions. So it's actually shifting the preference for conformations inside of the complex. Now, to make it fast enough for other people to actually use these, we need to somehow make it possible to run this on some sort of reasonable timescale to do these calculations yourself. The hardware is going to get better on its own. People are continuing to optimize hardware for machine learning. We don't have to worry about that. But what's really slow is computing all of these features is really difficult. That machine learning frameworks are not meant to do like molecular mechanics style pair lists and all of the calculations that otherwise slow down these calculations. So we've worked to optimize these and get rid of some of these bottlenecks through this library called NMROps, which comes from Gianni De Fabridis and Peter Eastman, the OpenMM developer. And what's cool about this is that they've been able to optimize this enough so that now with OpenMM 8, you can install this in a single line and actually get speedups that are huge compared to trying to just run this through the normal PyTorch. In fact, you can run an ML-MM ligand and protein simulation at something like 100 nanoseconds per day with one of the Annie ensemble members compared to about 600 nanoseconds a day with just running MM on the GPU. So it only slows it down by a factor of 5 or 6. Half of that is because you have to take a 2 femtosecond time step. You can't use constraints anymore in modeling these simulations because the quantum chemistry is really not meant to be used with constraints. And the other half of that comes from just the fact that you're doing a lot more math on the GPU. And so it's really easy to use. This is what you normally have to do to set up a protein ligand simulation using something like the open force field. And then all you have to do is say, I want to use any 2x, which is one of the very few general machine learning potentials on part of the system, and replace the ligand atom interactions with that inside of that region. Now, I don't think we're going to be using this for all that much longer, right? Treating only part of the system with something learnable, but using all of this molecular mechanics rubbish from before is really a difficult proposition to think that we're going to be stuck with that for another 20 years, right? So thinking about what is the future going to be like, where we can get rid of some of these low-order Taylor and Fourier expansions and replace them with something that has much more coupling to represent all of the quantum chemistry. And so something like Annie would be very useful, or the other new architectures that are coming out. There's a huge amount of innovation in this area. And what's really neat about these kinds of machine learning models is that they're often free of singularities. For some reason, we decided to build molecular mechanics models where they have singularities everywhere, which means that they're entirely numerically unstable. You have to do all of these very weird modifications to them that incur a huge amount of engineering effort if you want to do any kind of disappearing or inserting of atoms. But you don't have to worry about it at all with these machine learning potentials. They're singularity free. There's other issues you have to worry about, which we're finding. But the cool thing is we're trying to do a free energy calculation in a droplet of water. So everything here is treated with machine learning potentials. We're going to try to migrate this proton from here to this nitrogen over here. Normally, I would not be able to do that in an MM potential because I don't have the covalent bond breaking and formation energies in my MM. It's just not able to do that. But what I can do is I can combine them linearly. I can go back to the earliest days of free energy calculations and linearly interpolate between two Hamiltonians to give me a singularity-free way of computing the free energy of turning that off and turning another atom over here on in that other potential. And I can do that in a way I can take the derivative. I can take the derivative of all the parameters with respect to the free energy. And that allows me to say, use some of the physical free energy data I have. In this case, it's tautomer free energy, so it's not a super exciting protein ligand binding free energy, but it's a glimpse of what things will look like in the future when we can use a small subset of free energies to improve the accuracy reducing the error from an embarrassing 7 kcals per mole to still an embarrassing 3 kcals per mole. But it's a start, certainly, at being able to use data to improve accuracy of future predictions. And so I think this is going to be one of the really key ways in which we use statistical mechanics and learnability in order to use the data that we're getting from projects. Now, what I mean by that is, if you're doing a drug discovery program and generating all of this data with a huge amount of money and effort that's been spent on this, you come to work the next week and you have to use the same model again. Unless someone decides to update your force field in the meantime, it's just not going to get better using that data you spent time to collect. So I'm envisioning this future where we can take all of that data, build the model over the weekend by tailoring our force field, either MM or MLMM or machine learning force field ideally in the future, and being able to use that new, more accurate model, given the data that you've generated, to generate results the next week. It's really important, though, that you realize, we're not in a big data regime here at all, where, DALI was trained on something like 650 million images, and GPT-3 was trained on 22.5 billion pages of text. So if you consider the fact that you might make 2,000 compounds in your discovery program, it's like comparing a single book to the entire Library of Congress, right? There's a very different scale that we have to work with than the machine learners. So we really have to think of these extremely data efficient methods in order to make progress, which is why it's really important that we use all of the physical invariances and physical laws that we have, physical models, and then paste in the machine learning only where they're needed. And so what would be great is to work towards this idea of not only learnable potentials, but learnable protocols for these alchemical free energy calculations, adaptive and learnable ways of staging these networks of transformations, and then ultimately fusing them with these very fast equivariant structure-based machine learning models to prioritize which calculations should actually be executed at this very high level, high expense level of theory, which is why I'm super excited to say that, as Gerhard hinted at, we've had a prolongation of our Einstein collaboration with Andrea Volkamer, and we have a very exciting opportunity to hire 2 new postdocs to think about how to build this integrated approach of, now including, fantastic new things like OpenFold, which is able to predict different structures. You can use templates or potentially condition on other states in order to generate different conformations, fuse that with things like ML docking or advanced ways of modeling ligand binding, and then feed that to these structure-based machine learning methods and free energy calculations in order to actually predict these properties of kinase inhibition, selectivity, affinity, and mutant impacts in a way that's actually going to be useful for designing new therapeutics. So with that, I've spoken for too long, and so I'm going to say thank you and happy to take any questions.
John, thanks very much for this really nice and very great talk. So we are going to take questions both from the chat, from the audience, and also you can just basically speak up if you want to take a question from the Zoom part. So are there any questions from on site? Yeah, Catherine. But you need the microphone.
Hi, yeah, thank you very much for this talk. It was very interesting. I would be interested whether you already tested this hybrid method of physical and machine learning models in predicting bioactive conformations of molecules, and if it worked or
Bioactive conformations of molecules, like how does it actually bind in the binding site?
Yeah, or I mean, there are always different possibilities on how a molecule can adopt a conformation, and I'm a pharmacist, so for me, the bioactive conformation is interesting. So I would be interested in that.
It's, there's different ways people talk about this. Sometimes folks are just interested in enumerating plausible conformers that could be docked poses, for example. Others are really interested in how it recognizes the particular binding site. The free energy calculations do give us a picture if you're doing a transformation from one reference compound where you have a crystal structure to another proposed compound. They give us an idea of what that equilibrium ensemble should look like. So we often extract and examine what we think the plausible, both the pose and the heterogeneity and the conformation of interaction actually looks like. There's some other folks using machine learning very effectively right now. In fact, you've got a Microsoft research site led by Frank Noe here in Berlin that's collaborating with Max Welling in Amsterdam and Marvin Segler in Cambridge A lot of them are working on pioneering ways to use things like diffusion models to predict molecular conformers in novel ways that could lead to bioactive conformations by learning on bioactive conformations from the protein data bank or other sources, for example. So I think it's going to be a really exciting time where these models really learn things in ways that you can condition potentially even on like GPT-3 perceived text or something like that. So you can tell it, I would like the bioactive conformation for this kind of thing, and it can generate it for you. So watch out for the next few years there. It's going to be exciting.
Okay, chat. Okay. So no chat question.
Hello, everyone. First, thank you very much for the talk. Quite interesting. And I think you clearly make the point that machine learning can improve many computational chemical tasks. But machine learning is not like machine learning. So how do you find out which kind of algorithm of the many different algorithms that exist do you actually need to find the best solution and the best predictions for your specific tasks?
That's a great question. And even for the statistical mechanical side of things, there's no real theory that allows you to always decide on the best thing, right? So you essentially need to find something that is useful for your task, and that might change if the data distribution changes as well, or if the behavior of things. So often we're interested not with finding the best, but something that is better than we have in place right now, which is often a lot of humans getting together on a Zoom call and making proposals about what molecules to make, right? So by putting in models that have some predictive capability and then evaluating two models against each other to see which one seems to be at least retrospectively doing better and then prospectively performing better, we can switch from one model to another. So in our antiviral center, for example, we're sometimes running some of the models head to head to see which one is performing better, and then we can switch to that one, for example, if it's delivering better results. It's very hard to know which is the best model. There's this whole field in machine learning where everybody tries to come up with the new state-of-the-art model where your number is slightly better than somebody else's number, but then there's no quantification of statistical certainty or confidence in that result. So you really don't even know if the number differences are meaningful. And so I think there's a statistical issue with how that's done in the machine learning field right now that has to meld with how people are actually doing these statistically reasonable, robust analyses and comparisons between methods in computer-aided drug discovery right now.
Okay, thank you very much. We have one more question from the chat. One important concern when generating ligand force fields is its compatibility with the protein force field in use, using similar models, charge, balance, etc. Are OpenFF force fields made to be compatible with any specific protein force field, like the AMBER force field 14, etc.?
Yes, and so the current generation of open force fields were designed to be used with the AMBER family of force fields. So they should be drop-in replacements for small molecule force fields, but still provide compatibility with everything else in AMBER because they come from the same origin, which is a predecessor of the GAF force fields. They use the same Leonard-Jones terms, and they have a compatible charge model as well. The new versions will co-optimize the protein and small molecule force field. So that's open force field 3 and beyond. It's parsley, sage, and rosemary is the first one. That will be the first generation that does this co-optimization to try to get better accuracy for small molecule binding affinities.
Okay, thank you very much. We have no further questions from the chat.
Okay. David, another one.
So I think I really like the whole open force field consortium idea. I also know that there's something called open free energy.
Yes.
So I'm not 100% sure how you're involved in there. To be honest, that's whatever. But maybe can you give an estimate how far away are we from getting something like open force field models, but for free energy calculations?
So the Open Free Energy effort was an industry-led, the Open Free Energy Consortium is an industry-led consortium where the real steering governing board is all industry. We have an academic technical advisory board that I'm contributing to, and also have been very effectively collaborating with the folks there on building some unified object models that allow more plug and play of different algorithms. So one of the first things that's supported is one of our free energy codes that we're refactoring into their API. They've been doing a great job there. So I think they're very effectively making progress and intend to ship something that's really useful this calendar year. So I would really follow their work closely. we're going to start testing it next month, which I'm very excited about.
So great, thanks. We are running a bit out of time, so I'd like to stop the discussion at that point. So there is still the possibility maybe to ask questions in the chat. We're going to collect them and forward them to John. But now I'd like to thank John for this great talk again and for the discussion.